Welcome back, everyone. It is February 24th, 2021. It is week, I'm not sure, of our Water, Wetlands, and Watersheds seminar. And this week we are fortunate to have with us Dr. Anna Armitage. Dr. Armitage is a community ecologist. Her research interests are around food webs and ecological restoration in coastal habitats. Her current research is seeking to identify ecologically successful restoration techniques in coastal wetlands, and also studying the causes and ecological consequences of native black mangrove expansion into the northern Gulf of Mexico salt marsh sort of matrix. And she completed her PhD in biology at UCLA and is currently a professor of marine biology at Texas A&M at Galveston. So Dr. Armitage, thank you so much for joining us via Zoom. I hope you found a good plumber and that everyone, of course, is well. Your family and friends are well in Texas after the crazy past 10 days or so. Thank you for joining us.
Yeah, thank you so much. So it's my pleasure to be here. And like David mentioned, we had this major weather event in Texas last week. So this talk is really all pre-weather event, but I will end with some questions about where do we go from here, because that weather event did affect this ecosystem as well as many of our personal lives. So I'm going to go ahead and share my screen and start my presentation.
Great, go for it.
All right. All right, so I'm going to give you an overview today of some of the work my group has been doing to look at how, what are the ecological consequences of black mangrove expansion into Gulf of Mexico salt marshes. There we go. All right, so if you think about mangroves at all, you probably picture something like this, like these lush tropical forests that have really tall trees. In this lower right-hand corner, there's a couple of mangrove scientists for scale. You might picture these peaceful channels for kayaking down, these complex aerial root structures. And this indeed is what tropical mangrove forests look like. And in fact, that's what they look like in the southern part of Florida. But that's not the kind of mangrove forest I'm going to be talking about today. The type of mangrove forest I work in is something more like this, where you have these relatively small shrubs that are intermixed with expanses of salt marsh vegetation. So this is what the mangroves look like on the much of the Gulf of Mexico coastline. So taking a step back, mangroves are widely distributed on tropical and subtropical coastlines, indicated by the red by the red areas on this map here. Okay. And then salt marshes are largely restricted to the colder northern and southern latitudes, indicated by the black. But there are some areas around the world where you do get a coexistence of both marsh and mangrove vegetation, including parts of Australia, parts of South Africa, and the Gulf of Mexico, which is where I will be talking about today. So in this region, we have a coexistence of marsh and mangrove vegetation. So these Gulf of Mexico wetlands are transitional between marsh and mangrove. So what's the difference between them? Okay. Marshes are dominated wetlands that are dominated by short stature vegetation like grasses, sedges, and rushes. There are some short stature shrubs, but overall marshes are relatively low stature. And again, here are some wetland scientists for scale. Contrast that to mangroves. Mangroves are salt tolerant trees, and in the tropics, they can be 20 or 30 meters tall. On the Gulf Coast, like in Texas, they tend to be more like 1 to 3 meters tall. Here's my collaborator, Steve Pennings, for scale. They can be a little bit taller, but many of them are just about 1 meter tall, depending on the abiotic conditions. So even though that's not very tall, they still are taller in stature than most of the surrounding marsh vegetation. You can see some marsh grasses and succulents in these pictures, and they are much shorter than even these short mangroves. Another big difference between marsh and mangrove vegetation is that mangroves have aerial root structures as an adaptation to anoxic soil. And in the black mangroves we have on the Texas coast, these aerial root structures are these kind of pencil-like projections that extend out away from the tree and then stick up into the air. And what the consequence of that is that these aerial root structures convert non-vegetated habitat, like mudflats or tidal creeks, into structured vegetated habitat. So they really can alter the structure of that ecosystem. Now, largely the balance between marsh and mangrove vegetation is maintained because mangroves are more sensitive to freezing temperatures. And now and again, they will die back in response to freezing temperatures. And this allows marsh and mangrove vegetation to coexist. But that balance has been thrown into question in many places. And in many places, mangroves are expanding. And so areas that used to be mostly marsh vegetation are now starting to look more and more like solid stands of mangroves. And this has been documented in lots of places around the world where the mangrove distribution is moving north or south into cooler latitudes, depending on the hemisphere, or even in areas where they exist, those stands are becoming larger in size. And so this analysis from Kyle Kavanaugh from a few years ago shows change in the distribution or the size of mangrove stands on the Florida coast. And the larger the circle, the larger the change. These black circles are showing increases of 50 to 100% increase in area over a 10 to 20 year time period. In the southern parts of Florida, there's been a decrease in mangrove area indicated by these red circles. But you could see that in some of the, at the kind of northern range limit, there's been a large increase in mangrove area. And this kind of change is reflected in many parts of the Mangrove-marsh ecotone. And the consequence is that you end up displacing marsh plants. And because of those aerial root structures, you also get some conversion of mudflats into structured habitats. And this really changes the way the ecosystem looks. Now, one of those hot spots of expansion is on the Texas coast. This map is showing on the Texas coast, the coastal bend region, this little red square here in the center. And this is a blow up of that area around Port Aransas. Now, the colors in this map show change in vegetation from 1990 to 2010 over a 20-year time period. Now, these green areas are areas that were mangrove in 1990 and stayed mangrove in 2010. But all of these red areas here, over here on Mustang Island, over here in Redfish Bay, all of those red areas are areas that were marsh in 1990 and have been converted into mangroves since then. And so there has been this widespread expansion. This is just one example of mangroves in many parts of that ecotone. And many analyses and models predict that mangrove expansion rate may be accelerating. And some of those drivers include temperature. As winter temperatures get warmer, there are predictions that mangroves will increase in cover across much of their Gulf of Mexico range. This analysis by Mike Osland shows that these dark areas on the coast are areas where mangroves will be dominant by the year 2100 if winter temperatures continue their current trends. And similar outcomes will come from sea level rise, where you get increases in mangrove cover in areas that are susceptible to sea level rise. And these are just two of many potentially complex interactions that could influence the distribution of mangroves in this ecotone. So the bottom line is mangroves, the mangrove marsh ecotone is dynamic, and the distribution of mangroves in this ecotone is influenced by a wide range of biotic and abiotic factors. Some abiotic factors are relatively well understood, like winter temperature and sea level rise. Others, such as increasing nutrients and carbon dioxide, changes in precipitation, are less well understood. There are also abiotic controls that could influence mangrove distribution, including changes in pollinators or consumers, and those are also not well understood in this area. All right, so bottom line, mangroves are increasing in these ecotones and marshes are decreasing. But so what? What does this mean for the ecosystem processes, and functions. So our research team has been addressing this very broad question: which ecosystem processes and features are most affected by mangrove expansion? And then, based on that information, we hope to use it to try and inform if and where we should be using mangroves in restoration projects. Should we be continuing to use marsh vegetation in restoration projects, or should we be implementing mangroves? And so I'll touch on that question by the end of today. All right, so our group has been looking at lots of different elements. Today, I'm going to focus on a few specific aspects of mangrove expansion. Specifically, I'll talk about how it could change habitat characteristics for migratory, resident, and endangered species, such as the whooping crane, which winters in that hot spot of mangrove expansion. And then we'll talk about some of the potential trophic interactions that could be altered by mangrove expansion. I'll touch on some consequences for carbon sequestration, as well as erosion and shoreline stabilization. All right, so this talk is going to include information from a series of studies at sites across the Texas coast, primarily in the Port Aransas area and the Galveston Bay area on the upper coast. And in these maps, you could see the red teardrops are the mangrove areas, and the yellow arrows are the marsh areas. And we have sites at both the central and the upper coast. And I do want to point out that we also have this white teardrop here is a mangrove removal experiment. And so I wanted to touch on what that entails as well, because that's something that is unique to the Texas coast, where we are allowed to cut down mangroves, very different than in Florida. All right, so this large-scale mangrove removal experiment involves a series of 10 plots where we created a gradient of mangrove cover, ranging from 0% mangrove cover to 100% mangrove cover. And then we had these grids of mangrove, these kind of cells, to create that gradient of cover between those two extremes. And these plots are quite large. These plots are 24 by 42 meters in area. And if that sounds like a lot of work to create these plots, you'd be right. It was some of the toughest field work I'd ever done. And so what it involves is first measuring out the size of those plots, and the individual 3 by 3 meter cells that were either flagged for keeping the mangroves or removal of the mangroves. And then you give the postdoc a weed whacker with a buzzsaw attachment, and he cut down the mangroves that were in those cells marked for removal. And then you have the graduate students haul that cut material out and put it through a wood chipper on site. And then you let the marsh plants colonize in those cleared areas. And so within a couple of years, we had relatively natural mixtures of marsh vegetation in these cleared areas, a mixture of succulents and grasses. And we have been maintaining that project since 2012. Sometimes we go out and mechanically trim the mangroves to keep them constrained within their cells. And it also involves manual weeding to pull out any recruits. So we've been maintaining that experiment for the last 8+ years. All right, so what have we learned from this combination of survey and experimental approaches? So first, I'll talk about what happened with the marsh plants. So we used a series of kind of standard survey techniques to look at percent cover and species composition of the marsh plants. And the bottom line is that not just the cover of marsh plants, but also the species composition of marshes was different in sites with and without mangroves. So in this MDS plot, the black symbols are sites with mangroves over several different years. And don't worry about the years, because mostly there wasn't a big change among years. So the years were very similar to each other, but there was a big difference between sites with mangroves and sites without. So these marsh sites had no mangroves. And so the assemblages were quite distinct from each other. And that was largely because there were certain species, certain succulents, like some Sarcocornia, or grass species, like Disticlus, that were really very rare at sites with mangroves. They really only existed at those marsh sites. And so we saw not just differences in assemblages, but also decreased species richness as mangrove cover increased. And there was this threshold. Once you crossed about 50% mangrove cover, you saw a large decline in not just marsh cover, but also marsh species diversity. And this raises questions about the animals that are associated with these marsh plants and rely on them trophically. So does that decreased marsh plant cover have trophic implications for those animals that rely on the smooth cordgrass or other species in those marshes? And so the jury's still out on this particular question down here, and perhaps a year from now, I'll be able to give you an update. We are exploring this question. We also looked at the associated nekton and epifauna that were in these marsh and mangrove sites and saw some different results. This top panel here is an MDS plot for fish, and the bottom one is for invertebrates. And so the symbols and the colors are the same as in the previous graph. So first, let's look at what's going on with the fish. So the fish shows that there were differences in assemblages, but there was quite a bit more overlap between mangrove and marsh sites. So it wasn't quite as distinct as it was for the marsh plants. And there were a couple of species like, say, the bay anchovy that was much more common at marsh sites, but the fish assemblages were also quite variable, so there was more overlap. There also tended to be higher species richness of fish at those marsh sites. The invertebrates were a little bit more distinct, where you had more distinct communities in the marsh versus the mangrove sites. There was less overlap. And there were, again, certain species that were really only associated with marsh sites, certain grass, shrimp, and gastropods. So we didn't see a difference in species richness with invertebrates, but there was a more distinct difference in assemblages. We also looked at some of those higher consumers, the birds that use these wetlands, especially in the winter. And so to generate this analysis, we looked at a community-driven database of bird observations called eBird. And so eBird is a website where birders upload their observation lists. And if they check certain boxes, that means that their observation lists are quantitative. They record not just the identity of birds, but how many they saw and over what time period and at what specific sites. So we filtered the thousands of reports on eBird to select those sites that had these kind of analysis, research grade quality, and then selected those from a specific dates that were focused on overwintering seasons. And we divided this analysis into waders, which are herons and egrets, and then shorebirds, which are the sandpipers and their allies. So this graph is showing the shorebirds. So what the bottom line is here is that there were the shorebird species richness was higher at marsh sites, and the assemblages were distinct. There was very little overlap between marsh bird assemblages and mangrove bird assemblages. And largely this was driven by the presence of a few particular types of birds, dowitchers, yellowlegs, and certain sandpipers were common in the marsh sites, and other birds were more common in the mangrove sites like willets. Okay? So there were distinct assemblages and higher species richness at the marsh sites. Now, this figure shows shorebirds. We did the same analysis for waders, which are the herons and the egrets, and we didn't see a strong impact on wader assemblages between marsh and mangrove sites. But there's a little asterisk here, and that's because there was one really important difference, and that was the presence of whooping cranes. So whooping cranes are a highly endangered bird that that overwinters at one spot on the Texas coast, and that is pretty much its primary and sole wintering location. And in those marshes at the Aransas National Wildlife Refuge, there's also quite a bit of mangrove expansion. And so there's a lot of concern that mangrove expansion will have a disproportionately large effect on this highly endangered bird, which needs large areas of marsh to forage in the wintertime. So even though statistically there was no effect of mangrove presence on waders, there could be a very important ecological effect for this specific endangered species. Now, interestingly, we also saw that there were many fewer birdwatcher reports in areas with high mangrove cover. So not only were there fewer birds and less richness of birds, there were also fewer bird watchers in those areas. And so we don't really know why that is, because this wasn't this was sort of an observation from the data set, but we didn't actually survey the bird watchers to ask them why they did or did not go to particular sites. There was no difference in accessibility because all of the sites we selected were right off the side of a road. So whether the bird watchers avoided those sites because there were fewer birds or because the birds were harder to see because of the taller mangroves, it's hard to say. But regardless, bird watchers seem to be avoiding those sites with many mangroves. And this could have implications for the major bird watching industry, the economy around bird watching that much of this area is known for. And so there could be ecosystem service implications down the road, and that's another area for more quantitative exploration. Now, so those are differences in a summary of some of the key differences in community composition, but we also see some implications of mangrove expansion on trophic interactions. So I'm going to highlight for you just a couple of results from work that my students have done to look at some of the trophic consequences of mangrove expansion. So in this study, that looked at how the presence of mangroves altered predation success using blue crabs and one of their prey items, Penaeid shrimp. And this study was done in a Mesocosm where crabs were allowed to hunt these shrimp in areas with simulated mangrove vegetation or marsh vegetation or in open tanks. And this is showing time from the start to the end of each trial and the number of shrimp consumed on the y-axis. And what I really want to highlight here on this star is that in those tanks where there was mangrove vegetation, that rigid woody pneumatophores, those aerial root structures, it took the crabs a lot longer to find and consume that first shrimp. And then eventually they did consume shrimp, but they consumed fewer, and it took them longer to get those shrimp. So lower predation success, but just as much time and effort expended in foraging suggests that there may be a greater energetic cost for blue crabs to forage in mangroves. Now, this was a mesocosm study, but the student also did a correlative or a corollary study to look at predation success in the field using a natural set of predators, including both birds as well as crabs, and those results are in process, and hopefully I'll be able to share those the next time we talk. All right, another trophic consequence that could be important is for basal consumers. And there are a number of really important basal consumers that either directly consume marsh vegetation or consume the detritus produced by marsh plants. And so in this slide, I'll focus on fiddler crabs, which burrow in the mud around marsh and mangrove vegetation. and consume the detrital material produced by the surrounding plants. So in this study, we demonstrated that these fiddler crabs, when they are given choices between different types of plant detritus, they much prefer consuming the detritus of Spartina than the detritus of either Avicennia or plain sand. And in fact, they would just as soon sort through plain sand as consume any Avicennia or black mangrove detritus. So they preferred that marsh plant diet. And then we also saw that the diets of those mangrove leaves end up lowering crab as well as snail fitness. And so these papers are either in press or in progress. And so you'll see more about this coming up soon. But the bottom line is that mangroves are not a suitable or an ideal substitute for marsh vegetation. It's less preferred, and it can reduce the fitness of consumers if they have to eat it because there's nothing else to eat. All right. So moving away from the organismal consequences, I want to talk about some of the more ecosystem function consequences of mangrove expansion. And I'll touch on some of the potential for mangroves to influence carbon storage. So the sort of the general paradigm based on tropical mangroves is that mangroves will store more carbon in their biomass and also below ground than marsh plants will. And that is really just based on their size. Mangroves are bigger than marsh plants. But here in our ecotone, mangroves aren't that much bigger than marsh plants. So do they substantially still contribute, make a substantial increase to carbon storage in this ecotone? So to address this question, we went to that mangrove removal experiment and then divided the plots into the fringe area where the mangroves were larger and the interior area where mangroves tended to be a little shorter, and then looked at carbon storage, carbon pools in both the plant above and below ground, as well as in the soil. And so in terms of the carbon storage in the plant biomass, indeed, mangrove biomass, that biomass does exceed marsh biomass. This is showing biomass in the mangrove and the marsh, in the fringe and the interior zone. So both in the fringe and in the interior, mangroves are higher than in the marsh. And this is true for both above ground and for below ground. And this directly correlates with the amount of carbon stored in the biomass. So mangroves do have higher biomass, carbon storage in their biomass. But does that mean that more carbon is being stored in the soil? And in fact, it does not. So when you look at the surface soil carbon content in mangrove and marsh plots in both the fringe and the interior zone, you'll see that it's very similar. There's very little change in the surface soil carbon across these different vegetation or elevation gradients. So why is this? So why isn't that higher biomass of mangroves increasing carbon storage in the soil? Well, it's really a function of productivity. And in this region, below ground productivity, which is like turnover of the tissue, is actually pretty similar between marsh and mangrove vegetation. So even though marsh biomass is lower, the marsh biomass turnover or productivity rates are relatively similar, especially because our mangroves really just aren't that big. And so even though they're bigger than marsh plants, they're not so big that they can make up for that really high growth and productivity and turnover in the marsh plants. And so the jury's still out on whether mangroves actually improve carbon storage or sequestration potential on the Texas coast. It might be more of an even exchange rather than a real magnitude of benefit. All right, and then the last ecosystem process I wanted to talk about is erosion. So one of the reasons why mangroves are considered to potentially be a net ecosystem benefit is that they may be better at stabilizing shorelines and preventing coastal erosion. And so we had this unexpected opportunity to test that. we go. There's my little graphic. In 2017, when Hurricane Harvey came ashore as a Category 4 storm directly over those study plots. And there it came ashore. And then a couple of days later, this is when it started to dump those huge amounts of rain on Houston. So while we were in Houston, totally rained out, we got on the phone and developed a plan so that we could get back down there and see what happened to the erosion in these plots. So after the storm passed through, we went back down to Port Aransas, dusted ourselves off, cleaned ourselves up. And then we started to quantify the erosion that had occurred in plots with lots of mangroves and plots with few mangroves. So what did we find in terms of erosion? Basically, where there were no mangroves, there was a lot more soil loss. So this is showing change in elevation, so vertical erosion, in the along that gradient of mangrove cover from 0 to 100%. And you can see that in 2016, there had been, this is pre-storm, there had been a lot of loss, but there was a big increase in loss after the storm in 2017. So Hurricane Harvey caused more erosion in plots that had fewer mangroves, especially there was a lower magnitude of erosion loss in plots with some mangroves, but once you got to be above about 45% mangrove cover, there was really no hurricane-induced erosion. So it was really in areas with fewer mangroves where you got more hurricane erosion. And a similar pattern emerged for edge erosion. So not vertical, but horizontal erosion, where the edge of the plots retreated. Originally, the edge of the plots had been out here on the outside of this stump of a mangrove we had removed. But in those plots with fewer mangroves, the shoreline retreated several meters, up to 9 or 10 meters in some cases. And so there was more hurricane erosion in plots that had less mangrove cover. So mangroves were more effective, at least right in the eye of the storm, at stabilizing the shoreline and reducing erosion. All right, so where are we with all of this? Where are we so far? So these ecosystem studies have suggested that mangroves reduce vascular plant richness of marsh species, which may have consequences for altering carbon supply to herbivores and detritivores. And we have seen that mangroves also alter fish, invertebrate, and shorebird assemblages with a general pattern of a decrease in richness and abundance. And this may decrease the recreational value of these coastal wetlands for hunters, fishers, and bird watchers. We saw some trophic implications. Mangroves are altering predation dynamics, reducing basal consumer fitness. And so these studies are ongoing to expand the scope and the number of species involved. We do not see that mangroves substantially add to the soil carbon pool, at least on the Texas coast. And this conclusion is different than in many other areas where you see mangroves, in part because the mangroves in Texas are relatively small and have similar productivity in these wetlands. But we did see that mangroves were better at reducing coastal erosion. So where does this leave us? So like many parts of many coastlines around the world, many coastal wetlands are experiencing widespread habitat loss, as well as shifts from mangrove to marsh vegetation. So you put together this habitat loss, and there's this continued need to try and manage and maintain and restore these coastal habitats. And these images here are showing some of those types of changes that are occurring on the Texas coast. The upper image is showing an area on the central Texas coast where all of these red areas, and here are areas that used to be marsh and now are mangrove. And then this lower image is showing a part of the lower Texas coast near Padre Island. And these blue colors in here are areas that used to be marsh, but have since been submerged by sea level rise and are now open water habitat. So there's a number of different drivers of habitat loss, both biotic and abiotic, human and non-human linked. But in order to maintain all of the important functions associated with coastal wetlands, there's this continued need for management and restoration. So with that in mind, should we be using mangroves in restoration projects in Texas or other parts of the mangrove marsh ecotone? So my recommendations here are based on what we know and what we don't know about the consequences of mangrove expansion into coastal wetlands. Now, those small individuals of black mangroves, Avicennia germinans, they tend to have relatively minor effects on the surrounding flora and fauna. So their effects of small individuals are really localized to that immediate vicinity of that shrub or tree. But as those shrubs increase in size and the canopy starts to close, this sphere of influence increases in size and magnitude. So initially, putting mangroves in restoration sites will have pretty much no effect on the ecosystem function. But ideally, if you're putting mangroves in restoration projects, they will eventually grow and close the canopy, and you will end up with a very different kind of ecosystem. So what this bulk of studies shows is that mangrove and marsh functions are not interchangeable. There are some things that are better in mangroves or have higher functions, and other things that may be better in marshes, but they're not equivalent. And whether you define it as better or worse really depends on what your goal of that restoration project is. So if you want to maximize the ecosystem benefits of using mangroves in restoration, you should focus on those areas where mangroves are already present. So rather than transporting mangroves into new areas, if an area that has been lost is dominated by marsh, then that should be the focus. We shouldn't be shifting our plant focus to something new. Instead, we should be focusing on what the current plants are in those areas. And so many restoration projects in the Galveston Bay area continue to focus on smooth cordgrass, Spartina alterniflora. These projects help stabilize shorelines, also provide great outreach opportunities to get increased stewardship and involvement of local communities. All right, so all that being said, this talk was obviously, well, maybe not obviously, but this talk was written before last week's major weather event. This is a view out of the front of my house where we had snow on the streets, ice on all our plants. This is a picture from my yard, not from the mangroves, but similar conditions were experienced down in on the coast across the Texas coastline. And so what does this mean for all of these ecosystem differences in ecosystem functions between marsh and mangrove vegetation? These are a couple of our long-term monitoring plots taken before and after a freeze event. And so immediately after a freeze event, most of the above ground tissue will brown up and drop off. But the real question is, are these plants really dead, or are they going to regrow from the base? And past studies suggest that they are likely to regrow from the base, but this past event was pretty cold. It's pretty close to that threshold of potential mortality. So time will tell if we end up redeveloping these mangrove forests, or if we end up doing sort of an ecosystem reset back to the marsh conditions. So all of that stuff we just talked about really now needs to be assessed again in the context of this major disturbance event that's affected the Texas coast. So with that, I'd like to just conclude by acknowledging that this work was not done in a vacuum. Steve Pennings and John Kamenowski are two of my partners in crime here, and a whole bunch of amazing graduate students and technicians and undergraduates who have assisted along the way and have done a lot of this as part of their own research program. Lots of partners and funders to thank as well. So with that, that's all I have for today, and I'm happy to take any questions. Thank you.
Thanks, Anna. Thank you so much for sharing with us. That's quite a big quadrat you had in the last view there. It must be 2 meter by 2 meter. Sounds tremendously annoying, but yeah. And so I think, a lot of us think about the, conversion process and the sort of slow march, but looking across all of those services or functions, And inherently, we do sort of think of them as sort of more good or bad, or benefiting human society in some way. So that was really a really awesome sort of smorgasbord of services. We'll go back and forth with questions. If I'll start with one from YouTube, and then anyone in the Zoom room who wants to ask. Here's one from Ben Solman. He asks, says, great talk. Is it possible that the strength of mangrove impacts on soil carbon will change over time as climate and sea level change?
Yeah, so that's a great question, and it's because there's a difference between soil carbon pools and real potential for storage, for like decadal level storage. And you're right, that's going to be influenced by things like sea level. And so in these areas that are vulnerable to sea level, it may be that erosion basically releases carbon that's been stored in those sediments. So that's beyond the scope of what we addressed in that particular study, but I think that's an important consideration in, say, parameterizing or monetizing a blue carbon market, where people are trying to maybe conserve habitat to offset carbon production elsewhere. So that interaction with sea level rise is a very important consideration, but I don't have a quantitative answer for it about it.
Okay, thanks. Well, I'm going to follow up on that one with one of my own, which is you looked at those soil carbon numbers, found them to be the equivalent between the two patch types. But is there a function of time in there? In other words, like the mangroves are, they pop on, they pop off, but the marsh has accumulated carbon for longer. Is it possible there's a mix of carbon sources within that soil that is not accounted for, or that given longer to catch up, that the mangroves would knock it out of the park, or
Yeah, that's a good question. My guess, my best answer for that right now is that what I showed in that figure is actually surface carbon storage, and so it's most likely to reflect the current plant community. You'd more likely see those kind of that mixture as you go deeper down. And so this was really looking at surface carbon storage. We have not yet gone down deeper to look at kind of the history of the site, because then you would get more mixing. And that's an interesting question. A little harder to address in this experimental site where we remove the mangroves and then let marsh plants grow back in. So we're really looking at the current influence of the current plant community.
Right, and it's hard to necessarily track, like in a year-to-year sense, the carbon accumulation from that year, although I suppose there could be ways to do that as well.
Yeah, we did do some feldspar plots where we put down feldspar and looked at the accretion over that time. And so that was part of how we documented that this was stuff that had accumulated over a certain time period, but it's relatively recent, right? We have not looked down to look at more historical time changes over time.
Great. Any questions from those in the Zoom? Here, Mark, I see you unmuted.
Yeah, I've got a question. You talked about in your experimental plots, you went in and removed the mangroves and then allowed the salt marsh species to come back in. And it sounded like maybe 2 years-ish was a kind of recruitment time for that. And I guess what I'm thinking about now is this event that you've just experienced out there, and this idea of resilience within the community, and the fact that salt marshes can tolerate that, mangroves can't. So let's just say you have a major die-off with the mangroves, and how the idea of recruitment at a large scale where you might, generally I think of a lot of salt marsh species moving in through vegetative growth, having some sort of a propagule nearby that can fill in a gap, and not so much through seed recruitment because the germination isn't that great. So I'm just kind of curious what your thoughts are, and at your experimental plot scale, maybe that die-off may not have as big a signature for recruitment as if you were in your area that's like 100% mangrove that could have gotten completely killed off in its recovery time as a marsh now. And just kind of open thoughts as you move forward in this kind of next phase of the unplanned experiment.
Yeah, those are excellent questions, and they're questions we've been asking ourselves as well, because if this mortality event is really just above ground mortality, then it's really just a pause on the mangrove expansion, not a reset. But if there was complete mangrove mortality, then you're right. The patterns of succession are going to be influenced by the neighboring plants, as well as those seedling recruitment events from externally. And so we're hoping to explore some of those questions over the next couple of years in these in this great in that mangrove density study. So it's an unplanned natural experiment, but I think it would give us some interesting insights to the influence of succession on the future condition of these coastal wetlands.
Yeah, that's great. We'll look forward to the results of that.
Yeah.
Okay, I was muted. Okay. Going back to the YouTube, a question following up again on carbon. So we were talking about productivity differences, but AJ Reisinger asked, what about soil respiration? So are there differences in litter quality, for example, that might drive differences in respiration rates and therefore the carbon storage that you saw?
Oh, interesting question. So my collaborator, John Kamenowski, has been exploring that. And we have not yet been able to really pull out those differences in respiration that's linked with particular types of litter, in part because we've done litter incubations and then the crabs get in them and rip them all apart. And so we've had some sort of methodological challenges to try and answer that question. But we do think that there could be differences in bacterial activity or microbial activity that could influence these decomposition rates. We've just not been able to quantify it yet. So that's a good question and an area that we've been trying to get a little further in with. So stay tuned, I guess, for that.
And we have one more from Elliot White. It was back to the original, or some of the earlier slides with you showing the NMDS plots and things like that. And he asked, have you done cluster analysis to see if those are statistically significant or insignificant groupings?
Yeah.
Because, there was, the PCAs and all that stuff, they visually look different. And so how do you decide whether they're significant? Does it, and maybe just philosophically, does it matter to your interpretation?
Yeah, so we didn't do cluster analysis because we had a priori expectations about differences between mangrove and marsh, but we did do analysis of similarity and permanova, so permutational ANOVAs. And so those that I talked about, if I talked about them being different, they had statistically significant differences. So I didn't present the statistical results in this talk, but if I maybe you remember, maybe you don't, but for the waders, I didn't present a graph of the waders because there was no statistical difference, but there's still this really important consequence for a high profile, highly endangered bird. And so sometimes those differences between statistical and ecological significance are relevant. So the ones I talked about though are statistically significant.
I think we need to write that as a template for all response to reviewers from now on. Yeah. Andrew, I see that you unmuted. Do you have a question for Anna?
Yeah, sure, Anna. Anna, it was a nice talk. Thanks for sharing this, this body of work. I mean, it's really impressive. The scale of the experiments and then how thorough you've been with looking at the responses. It was really cool to see that.
Thank you.
And I know that it's ongoing. It sounds like there's a lot you're continuing to do, so that's cool. I'm just curious, early on in the talk, you presented what was kind of an overhead shot, probably like satellite imagery or whatever, and you had marked a kind of mangrove sites and marsh sites. And it looked to me like the marsh sites were kind of in the interior, of the bay, and the mangrove sites tend to be on the exterior, what was maybe a barrier island in that system. Yeah. And I was just curious if you could talk a bit about whether that's reflective of large scale physical factors, or if it's a dispersal limitation thing, or just sort of what what you kind of see happening there over the upcoming decades or something, assuming that this freeze doesn't completely change things or something.
Yeah, so that's an excellent point. You're right that there is this spatial separation between where the mangroves are and where the marshes are, especially in that Port Aransas area. And that's not something we can control. So we had to kind of acknowledge that spatial artifact when we're looking at especially things like the fauna. So the faunal composition could be very different in these internal areas that are calmer, less exposed to they have a more muted tidal exchange. The salinity regime is a little different. But that's also why we did the mangrove removal experiment as a partner to try and understand whether these patterns we were seeing at the survey sites, whether they were reflected in the removal experiment as well, which also has its own set of artifacts. And often there was quite good agreement in terms of things like bird use and marsh plant species richness that correlated quite well with between the experimental and the survey sites. Other things are a little harder to parse out, like the fish and the shrimp. Those are probably more closely related to the subtidal habitat that's adjacent to those wetland habitats than the vegetation itself. And that's why we're starting to look now at those trophic interactions, those species, those individual level interactions, to see how, when those animals are present, what does it mean for them to interact with these different types of plants? So yeah, so we're using a variety of different approaches to try and kind of address and acknowledge that consequence.
Yeah, you gotta get that. I mean, I guess I was more asking, like, what do you think it tells you about the fact that the marshes are in the interior? Like, is that telling you something about, like, I mean, I guess like the most basic question is, do you predict those will become mangrove? And the fact that they're not yet, is it because there's a different like thermal regime there, or is it a difference in just kind of propagule dispersal, or like, what do you?
Yeah, so my main guess is that it's dispersal limitation. And so as these mangroves are dispersed through these propagules that float, and there's some evidence that major storm events, for example, can cause big rafts of propagules to float and then settle in new areas. And that's sort of how these coastal areas experience recruitment events, is following these major storm events. And so most of the recruitment has been on the barrier islands. And so there is some dispersal limitation then to prevent those mangroves from getting into the inner bays, but they are there and they are becoming more common there. So they're more patchy, and a lot of it's on private land. So it's a lot harder for us to access, but we are starting to see mangroves in those inner areas. So I think now, before this week, I would have said it's a matter of time before mangroves become very common in those areas. We don't know how they were affected by the freeze. This may have kind of slowed that colonization event a little bit. So time will tell there.
Cool. All right. Thank you.
Sure.
Well, thanks. Let's give Dr. Armitage a round of applause. We appreciate you giving our wetland seminar today. It will be available on YouTube for forever. So you can always go and watch it. Thanks so much for everyone. Next week we have coming in, who knows what's coming next week? I always forget to bring this up. We have seminars. Next week we're going to hear from Heide Diefendorfer, applying cumulative effects to strategically advance large-scale ecosystem restoration. So please tune in next week. Thanks again, Anna. It was nice to meet you online, and we'll see